In this video lecture, I will be talking on tumor immunology. In 1909, the German physician Paul Ehrlich hypothesized that tumor cell can be recognized as foreign and eliminated by the immune system, and the recognition or detection of tumor cells is known as tumor immunosurveillance. So, tumor cell is a self cell. I mean by self, it is not foreign cell that undergoes some alteration. For example, expression of some tumor specific antigens, which is present only in tumor cells, or it expresses tumor associated antigens, which is present in both tumor cells and normal cells. Due to expression of these antigens, tumor cell will be recognized as non-self by the immune system, and they will become target for killing by two cells, CD8 or T cytotoxic cell and natural killer cells. So, first, tumor cells are target for killing by CD8. As can be seen here, this is a tumor cell. They express tumor antigens, which are endogenous antigens, and they get expressed on the surface of tumor cell in association with major histocompatibility class 1. So CD8 or T cytotoxic will recognize through their TCR the antigen, tumor antigen, and the major histocompatibility class 1 and get activated. Upon their activation, they produce perforin, which induces or lead to formation of some perforation on the surface of tumor cells, and this will allow granzymes from CD8 to enter to the cell, and it cut the nucleus into small pieces. So tumor cell in this mechanism of killing will die by apoptosis. However, tumor cells, they have some survival strategies to escape killing by CD8 through down regulation of major histocompatibility class 1, and this will lead also to down regulation of endogenously produced antigens. As can be seen here, this is a normal. Here this is endogenous antigen produced by the tumor cell in association with major histocompatibility class 1. But in order to escape killing by CD8, tumor cells, they evade killing immune responses by down regulating major expression of major histocompatibility class 1. So it will not be recognized as foreign or non-self. But however, sorry, low expression of major histocompatibility class 1 on surface of tumor cell will be sensed by another cell known as natural killer cell. And as can be seen here, this cell actually normal cell, it is not a target, not tumor cell. So this is a major histocompatibility class 1. So through the inhibitory receptors of natural killer cells, the natural killer cell will not attack this cell. But however, when tumor cells downregulate expression of major histocompatibility class 1, this will be sensed by the natural killer cell through their inhibitory receptors, and in turn natural killer cell will produce perforin and granzymes which induce killing of tumor cells by apoptosis. Also, anti-tumor activity macrophages, M1 macrophages, they have capacity to kill tumor cells through their cytokines, interferon gamma, tumor necrosis factor alpha, interleukin number 12, and some of chemical intermediates, for example, nitric oxide synthase and reactive oxygen species which have anti-tumor activity. Nitric oxide synthase and reactive oxygen species are toxic to tumor cell. And as you may know that macrophages, they express Fc gamma R1 and Fc gamma R3 receptors, and these are specific for IgG antibodies. So when gamma globulins or immunoglobulin G capture tumor cell, so it binds to their specific receptor on surface of macrophages, and this will lead to activation of macrophages, which produce these cytokines, which have anti-tumor activity. And in addition to that, M1 macrophages have the function of engulfing target cells. Function of engulfing target cells. Phagocytosis, it is either phagocytosis, normal phagocytosis, or antibody dependent phagocytosis. Type 1 innate lymphoid cells, these are present in the tissue, and they release interferon gamma and tumor necrosis factor alpha, and also they have an anti-tumor activity, and they have the capacity to kill tumor cells. Thank you very much, Professor Ahmed Moulad. I would like also to thank Dr. Abir Abdelmunim for taking part in preparation to this lecture. Thank you again, Professor Ahmed Moulad.